Okay, so good morning, everybody. Thank you so much for coming. This is our last day of the Discovery to Cure. It's, for me, this mixed feelings in this day. One is, finally they're leaving. But no, it's just not kidding. They are leaving. And we're really sad because your children, you guys, always give us in the summer this special spirit and time. And second, to the parents, when they started here, I told them they are the evidence that evolution exists. Why? Because when they came, they were so shy. Are you still shy? They're not shy anymore. You will see by the videos. But moreover, it's amazing. We have been filming them. You will see that they're not shy anymore. But more important, when they came here, they were your children. They didn't know how to handle themselves. But more important, they didn't know even how to pronounce some of the scientific words. And you will see today, you will not recognize them after they present. Yeah. The Discovery to Cure has been running for several years, for 16 years. Not here. It's a program that we established at Yale University. And I was delighted to be able to move the program here to the Mott Center, to Wayne State and Detroit. The first year we couldn't run it because of the famous COVID. And this year, in spite of everything, we ran it, although in limited number. Usually we have between 25 to 30 students. This year we were running only with 10 high schools and 2 undergraduate students. We hope next year we will be able to grow back to the regional numbers. And you guys are ambassadors of the program. So I hope you will talk in your schools. You will make presentations at your schools. You will excite your colleagues, your friends to apply for next year. You are the first group of Detroit, of the Mott Center. You are our ambassadors. This program will not happen without the help of a lot of people. You know, for me, it's very comfortable. I'm sitting here, I'm talking, and you would think that I did everything. I did nothing. Yeah, really, this has been run by this young lady, this fantastic young lady, Alisa. And you will see around a lot of the people who are helping, taking the pictures. But there is another aspect that is extremely important. I want to really reiterate and thank especially the faculty and the mentors. Yeah. Without the faculty accepting, yes, let's give them a help. Without the faculty accepting to take students to the program, and the mentors, the members of the different labs that they spend a lot of time teaching the basic concepts, how to handle a pipette, how to put a tips and so on, you will not be here and doing what and the successful that you have done. So again, let's give an applause to the mentors and the faculty. Second, believe it or not, running this program, it costs a lot of money. A lot of money. Yeah, there is a lot of expenses that you have no idea that is behind the scenes. Even getting you that little thing, that card that allowed you to come in, and that costs money. Yeah, and by the way, don't tell anybody, although we are recording. I'm going to get in trouble. Keep it with yourself, okay? Yeah. I have that fight always at Yale, but I always tell them it's a good memory, so you keep it for yourself. Don't tell that I told you. But there is another aspect that is very important. You receive something, usually we give you at the end, at the ceremony, but because of technical problems, you receive a little present through the mail. Did you get that envelope? That's for your college fund. Yeah, not for parties, not for your college fund. Yeah, that money is coming from the National Institutes of Health. This program, the Discovery to Cure, is funded in part by the National Institute of Health, specifically the National Institute of Child Health Development. We submit a proposal, they love the plan, and they funded us. We have been funded already for 10 years. We stopped in the last 2 years, as I mentioned. We will have a renewal coming soon. Now that renewal will be associated with here. So your feedback is very important. You will be filling a form associated with what was your experience. Please fill up that form. For us, it's critical for the renewal of this NIH grant. I also want to take a special thank to the Ramos family. They initiated a foundation, an endowment that will be dedicated to support the discovery to cure. Yeah. And this, you have no idea how helpful is that, because with the NIH grants, we can cover some of the expenses, but there is many aspects that we are not allowed to cover with the federal funds. The help coming from the Ramos family is extremely important for us, and will allow us also to grow here in Detroit. So let's give an applause to that support from the Ramos family and the NIH. And also I want to thank my old friend, Dr. John Levy. I don't know if he is joining us today. But Dr. John Levy has been a supporter of the Discovery to Cure for many, many years. Yeah, at Yale as well as here in Detroit. What we hope once this pandemic passes, I'm talking too long. Yeah, I'm going to finish in a minute. Alisa is already looking at me. What we hope is that when the pandemic is over, we will be running parallel the Discovery to Cure here in Detroit, as well as the Discovery to Cure in New Haven, and exchange between the two institutions, between Wayne State and Yale. So with that, I'm going to stop the sharing. Oh, wait, I was sharing or was not yet? No, it's over there. Can you stop the sharing? It's here? No, it's not here. Oh no, wait, sorry. Technology. There you go. And what I would like to do is just to turn around this computer. And for those who are in Zoom,
say hi, guys.
Great. As you understand, because of the restrictions that we have, we are not allowed to have more than the number of people that you're here with the distance and the masks. With that, I pass it to the Master of Ceremony, Alyssa, who will start the program.
Thank you, Dr. Mor. Hi, everybody. I know all of you are excited for our students to present. So our first speaker today is from Regina High School, Emily.
All right, so as she said, my name is Emily Boyd. I'm from Regina High School. And this summer, I was given the project of working with ovarian cancer and understanding M6A modification. So as many know, ovarian cancer is a very deadly disease. In 2020, there were an estimated case amount of about 22,000. And in 2010 to 2016, less than 50,000 survived from it, while 80% within 5 years reoccurred. And overall, ovarian cancer is going to be characterized by metastases, which is what I'm focusing on in my project. And what causes metastases is epithelial to mesenchymal transition. So as we can see, an epithelial cell is going to be a normal cuboidal cell. And as it transitions, it's going to become a spindled cell that is able to migrate and invade and form tumors. So recently, AMT has been found to work with epigenetics. And as we can see, as an epithelial cell is becoming mesenchymal, it has the option to undergo epigenetic modifications. And for my project, I'm looking at RNA methylation. So RNA methylation is a post-transcriptional level of regulation. It's simply just the addition of a methyl group on an RNA. So in my project, I'm looking at M6A, which is just going to be an adenosine base with a methyl group added at the nitrogen 6 position. So M6A is an abundant eternal modification found in mRNA. It entails, as I said, methylation of adenosine base at nitrogen 6 position, and studies have demonstrated that M6A is reversible, and it is key to regulation of the RNA metabolism. So for this to occur, we have M6A writers. So these are going to be proteins that induce the methyl group. This can be METL3, METL14, or METL16. And then to demethylate, we're going to have proteins that remove the methyl group. These are considered M6A erasers, and that is FTO or ALKBH5. And then once methylation and demethylation has occurred, readers are going to come through, read the RNA codings, and carry out whatever has been coded for. So that can be like pre-mRNA splicing, RNA decay, or RNA stabilization. So this whole process has been indicated that EMT and epigenetics are important in cancer development. However, it hasn't been shown in ovarian cancer. So our objective is to investigate whether mesenchymal ovarian cancer cells have higher level of M6A compared to epithelial cells and its role in EMT. So our first hypothesis is whether mesenchymal ovarian cancer cells have higher M6A RNA modification than epithelial cells, which we're assuming will have higher writer and lower eraser. So our first question of our first experiment is, do mesenchymal cells have higher M6A RNA modification compared to epithelial cells? So to test this, we're going to conduct an RNA dot blot. To do this, we're going to do one epithelial cell line, R182, and two mesenchymal cell lines, F2 and R182. So to conduct the dot blot, we start with growing cells and feeding them. We're going to lyse the cells, separate the cells with chloroform, isolate the RNA and extract the pellet, and then suspend it in water to have RNA. And then with that RNA, we're going to dot it onto our membrane with 100 nanograms per dot. Expose it to UV to link the RNA to our membrane, and then incubate it in a primary and secondary antibody conjugated with HRP, and then we'll image it. So when we did this, this was our image. As we can see on the left, our epithelial cell, it's much lighter than our right. That means there's not as much M6A present as in mesenchymal cells. So we can conclude that with this, mesenchymal cells have higher M6A than epithelial cells. And then our second question, we're looking at if mesenchymal cells express higher M6A writers compared to epithelial cells. So our hypothesis is, do mesenchymal ovarian cancer cells have higher M6A writers compared to epithelial cells? And to test this, we're going to use qPCR to quantify the RNA expression level of M6A writers in our different cell lines. So for epithelial cell lines, we're using R182, R2615. And then for mesenchymal, we're doing F2, SKOV3, and OVCA433. So the writers we're going to be testing is METL3, which methylates RNA, METL14, which enhances METL3 binding, WTAP, which stabilizes METL3 and METL14. Those three are going to be found in the methyltransferase complex. They're going to work together. And then METL16 is an outside writer that methylates RNA and non-coding RNA. So when we conducted this, we can see METL3 using our epithelial cells as our control. Mesenchymal cell line F2 has almost 30 times more METL3 show up than both epithelial cell lines. And then METL14 has about 5 times more METL14 show up than our epithelial cell lines. And this is proving methylation is occurring because in the complex, they're bound together. And then WTAP, there's about 2 times more F2 shown than R182 and R2615. And then METL16, F2 has about 10 times more METL16 shown up than R182. So we can conclude that mesenchymal ovarian cancer cells do have higher expression of M6A writers when compared to epithelial cells. And then our last hypothesis is, do mesenchymal cells have lower level of M6A eraser compared to epithelial cells? So we're asking if the expression is lower of M6A eraser. So to test this, we're going to do qPCR again with the same cell lines, just different erasers. And then our result when using FTO, we see that FTO is demethylating more compared to mesenchymal cells, which validates our other research. And our conclusion is that mesenchymal ovarian cancer cells have lower expression of eraser compared to epithelial cells. So in summary, our mesenchymal cells have higher M6A modification compared to epithelial cells. This is supported by the higher expression of M6A writer showed in our qPCR, and that FTO is lower in mesenchymal cells compared to epithelial cells. And this data highlights the importance of further investigation on the role of EMT in ovarian cancer and allows us to inhibit ovarian cancer metastases. So in the future, we would prefer to immunoprecipitate M6A and sequence the RNA in the downstream genes that control M6A modification. So basically, we would fragment that RNA, expose it to M6A, and then get that sequenced and understand what other genes we need to be looking at to inhabit how the metastases are occurring. And then, thank you.
Our next presenter is Bao Boy from Parkway Christian High School. Bao.
Hi everyone, I'm Bao. I'm from Parkway Christian School. Thank you for coming again. So in my project, me and Candy, our overarching goal was to characterize HESC cells response to CpG. So CpG is our area of study. So before we get started with my presentation, I'm going to talk about a little bit about the key terms. So these are some terms that I think might be helpful to know. So we have human endometrial stromal cells, or in short, HESC and decidua. We also have CpG ODN, which is the treatment that we worked with. And I'm also going to talk about TLR9, cytokines, and CpG-PI complex. So again, our cell line is HESC, or human endometrial stromal cells. And as you can see here, this is the embryo where the baby is, and this is the decidua. So HESC cells are technically not part of the decidua, but after decidualization, it becomes decidua and endometrial stromal cells, which are part of the decidua. So you can see HESC cells over here. Talking a little bit about toll-like receptors. So they're a key element of the immune system, both innate and adaptive. Their role is to induce various cellular responses, including the transcription of several genes that later on leads to the elimination of pathogens like viruses and bacteria. So CpG, which is what we worked with, is recognized by TLR9, which is an intracellular receptor. So this is the TLR9 pathway in immune cells. So you can see here, after binding with CpG and undergoing phosphorylation, the binding of CpG to TLR9 could induce either interferon alpha right here, or pro-inflammatory cytokines like TNF alpha, IL-6, IL-8. So you might wonder, for those of you who do not know what cytokines are, so they are proteins that are released by the cells, and their job is to affect the interactions and communications between cells. So you can see in the picture over here, you have different cytokines and how they affect the communications of different immune cells. some of which we know kind of well, like B cells and T cells. So we're particularly interested in two kinds of cytokines, inflammatory cytokines and type 1 interferon cytokines. So inflammatory cytokines, like these three, are the ones that cause inflammation, which is the process by which your body removes pathogens. But you know from your doctors that too much inflammation could cause damages to your organs. So these cytokines need to be controlled by other cytokines. So that's when these cytokines come in. So for type 1 interferon cytokines, we have interferon alpha and interferon beta. They mostly, but not all the time, protect the host against pathogens without damaging the host. So now we're getting to CpG, which is what we worked with in our main area of study. So it stands for CpG oligodeoxynucleotides. It is a short single-stranded DNA, bacterial DNA, and CpG stands for 5' cytosine phosphate guanine and 3'. Again, it is recognized by TLR9, and the one that we worked with is CpG class B. So before we get into the data, I'm going to talk about PEI. So we added PEI to form the CpG PEI complex because as you can see over here, when you only add CpG to the cells, it only induces IL-6, but when you add the complex, it also induces interferon alpha. So that's why we used CpG PEI. So far we know that in immune cells, the NF-kappa B pathway, which you can see over here, is the dominant pathway in immune cells. But our hypothesis is that the interferon beta pathway, which is this, is the actual dominant pathway induced by CpG in HESC cells. So this is what we were trying to prove. So we designed experiment 1 to test whether or not our hypothesis is correct. So we treated HESC cells with 100 nanomolars and 400 nanomolars of CpG PEI. And then we did RNA extraction to obtain the RNA of the cells. And then we did qPCR to measure the RNA expressions of the genes that we want to test. So as you can see in the picture right here, there are a lot of dead cells in the 400 nanomolar treatment group. So from this, we conclude that 400 nanomolar CpG PEI is actually toxic for HESC cells. So these are our qPCR results. So as you can see right here, interferon beta is very upregulated, and this is our hypothesis. So these are the downstream interferon genes of interferon beta. So you have MIX1 and Viperin, they're both upregulated. So we suppose this could be the result of the upregulation of interferon beta. So, and these are the qPCR results of the inflammatory genes. So we have TNF alpha, IL-8, and IL-6, both upregulated, but when compared these genes to interferon beta, their upregulations are not as significant as what we saw previously. So our conclusion is 400 nanomolar CpG PEI is toxic for the cells, as we saw in the pictures. And we were able to validate our hypothesis that interferon beta is the dominant pathway in HESC cells. And we also know that the effects of CpG PEI on HESC cells are best observed at the 48 hours time point. So in our next couple experiments, our main goal was to verify the data that we got from the previous experiment. So this time we didn't treat the HESC cells with 400 nanomolar CpG PEI because we know that it is toxic for the cells. And we did the same thing. We did RNA extraction and qPCR. So as you can see in these pictures, the cells are doing fine because this time we didn't treat them with 400 nanomolar CpGs. These are our attempt to verify the data that we got from the previous experiment. So you can see, again, interferon beta is very upregulated by about 30 times. And as the result, MIX1 and Viperin were also induced by that. And when we get to the inflammatory genes, which are the downstream genes of the NF-kappa B, we could see again that they're upregulated, but not by that. So with this, we were able to verify that interferon beta is indeed the dominant pathway induced by CpG in HESC cells. So that is my project. So I want to spend a little bit of time to say thank you to Dr. Mor for his advice and inspiration, for correcting me for mispronouncing some of the scientific words. And most importantly, the opportunity to work in the Mor lab to gain so much knowledge about biomedical research. I thank Candy for her mentorship and her patience, as well as other people like Anna, Yuan, Hussein, Anthony, Andy for their insights and for their willingness to help me whenever I'm stuck and to answer my questions. I thank you, Alisa, for handling all the paperwork, and I thank Candy and Yuan for taking me to the lab and driving me home. So thanks, everyone.
Is there any question for?
Yeah. Are there any questions?
You don't have questions. You can ask. No? Okay, we'll let you go.
Thank you.
Our next presenter is Mariska Goswami from Troy High School. Mariska.
Hi, everyone. I'm Mariska Goswami, as Alyssa just introduced me, and I go to Troy High, and I've been working in Dr. Zhang's lab for the past 8 weeks on this project, which is identifying binding partners of the human DNA Li1 protein by yeast-2 hybrid screen. So to understand the DNA Li1 protein and its function, we can back up a little bit and review spermatogenesis. Spermatogenesis has 3 major steps, which are mitosis, meiosis, and spermiogenesis. And during this process, sperm cells undergo a lot of really important changes, including acrosome and tail formation, which is important because if sperm cells cannot grow tails and they cannot move, then they cannot fertilize the egg. Before I arrived at the lab, Dr. Zhang's lab had already found that the Parkin co-regulated gene protein, which is PACRG, and mouse meiosis expressed gene 1 protein, which is MEG1, form a complex that transport cargo important in sperm tail formation. And the complex is listed here. They also found, using the same technique that we did for this experiment, that PACRG interacts with DNA Li1, the focus protein of this project, which stands for dynein axonal light intermediate chain 1. Because they found that PACRG interacts with DNA Li1, they decided to generate DNA Li1 conditional knockout mice along with mice that have PACRG and MEG1 knocked out. Overall, the phenotypes were similar, except for a couple key differences in the DNA Li1 mutant mice that were not seen in the MEG1 PACRG mice. As you can see here in this photo, the 2 sperm cells have actually not separated and they have multiple tails as opposed to 1. And because this knockout caused such a difference in the phenotype, they hypothesized that DNA Li1 might have other functions besides the ones on the MEG1 PACRG pathway. Sorry, technical difficulties. I think it's working. Thank you, Alyssa. So yeah, I know. So from that, we decided that our goal was to explore potential new functions of DNA Li1 in sperm formation. In order to do this, we decided to perform a dihybrid library screening using yeast mating. And the way this works is it's a bait and prey situation. So our bait protein is DNA Li1 cloned in this vector, and our prey is a protein library that we've gotten from a company that's constructed in another vector. The vector that contains DNA Li1 is kanamycin resistant, and kanamycin is an antibiotic. And the protein library containing plasmid is ampicillin resistant, which is a different antibiotic. So if you tried putting the DNA Li1 plasmid in bacteria on an ampicillin plate, it would not grow. And same with the protein library and kanamycin. However, if we mated them and plated them on an ampicillin plate, then you would see growth if they mated correctly, because they confer both resistance to both antibiotics. In order to do this, the steps are to mate the bait and the prey, plate the yeast on the medium, isolate the DNA from that, and transform it to bacteria. And if it grows, then we know that they mated. So that's the screening process that occurs. More technical difficulties.
You have to click back.
Thank you, Alyssa, again. So before that happens, there are a lot of primary preliminary steps that go into before we mate the yeast. First, before I joined the lab, Dr. Zhang's lab actually designed the primer that amplifies the human DNA Li1 cDNA, and they performed PCR with it. That's the leftmost picture. The second well only, the first well was for a different experiment, but you can see that PCR happened correctly because the correct band links are there. And then they extracted that from the gel and performed TA cloning with it, and then sent it in for sequencing. So some of the inserts that they sent in for sequencing actually had no mutations, but I wanted to point out this one because it has one mutation, but is on the third base pair of the amino acid that it codes for. And sometimes amino acids, 3 base pairs code for the same amino acid, even if the third pair is different. So this mutation wouldn't have mattered, and we could have still used this sequence, but in the end, we decided to go with the ones that did not have mutations. After that, when I joined the lab, we performed subcloning with the confirmed DNA Li1 insert and our vector, the one that confers kanamycin resistance. And we transformed that to bacteria and then isolated the plasmid from it and ran this gel. In lane 1, you can see that the correct insert is not there, but in lanes 2 through 6, the correct insert is seen relative to the latter. We decided to use plasmids 3 and 4 for the experiment. We also had to test for autoactivation because the way the screening process works, if the yeast grows without being mated, then we will never know whether it has mated or whether it grows on its own. So the top plate is grown on SD-3 medium, which means that it doesn't contain 3 of the essential amino acids that yeast needs to grow. And if we plate the yeast with the plasmid on this medium, but not mated with the protein library plasmid, and it grows, then we know we can't use this for screening because it grew without mating. However, as you can see on the top 2 plates, no growth has occurred. To grow the yeast before we can mate it, we use SD minus tryptophan, which is just 1 amino acid that yeast need to grow. And tryptophan is synthesized by the plasmid that we transformed onto the yeast. But the AH109 yeast strain that we used cannot synthesize tryptophan by itself. So in order for it to grow, you can see that on the bottom left and bottom right, plasmids 3 and 4, there are growth on those plates, but not in the bottom center plate because it has not been transformed with the plasmid and therefore cannot grow on a medium that does not have tryptophan. Once we grow our yeast, we need to perform a Western blot in order to see whether our protein is being expressed. We used an antibody for a tag protein that is on the same plasmid as DNA Li1 called c-Myc, And the molecular weight of DNA Li1 plus the tag protein plus a little bit of the rest of the plasmid is about 47 kilodaltons. So we would expect to see a band right underneath 50, which we do see in all of our samples, but not in the negative control. If we had seen it in the negative control, it would have been possibly a non-specific protein, and we wouldn't have known whether our protein was being expressed. So with that confirmed, we could move on to mating and plating. In order to mate the yeast with the protein library strain, we need to grow it to an optical density of 0.8. Now, up until yesterday, the yeast had not grown past an optical density of 0.15. So this is where it gets a bit complicated. I took this picture yesterday at noon and right before I sent my presentation to Alyssa, and the yeast is now growing. Now it's mating as we speak in the lab upstairs, but because of these complications with growing the yeast, we couldn't finish the project before the Discovery to Cure program ended. So I will walk through the rest of the steps for the sake of showing what we hope will happen, but the rest of this is speculation as of right now. So we grew the yeast, we will grow the yeast, and right now it's mating. What happens is we take the yeast that has grown and we mate it with the protein library strain, which is very concentrated. We don't need to grow that. And then we shake it in liquid medium for 24 hours. Then we put a little sample of it under a microscope to check for a zygote, and a zygote will be seen when the yeast has mated. So this is an example picture. And once we see that mating has happened, and then we plate on the plates, the SD-3 medium that I was talking about earlier, that do not contain 3 of the essential amino acids, but we're also plating with this X-alpha-gal indicator. And what this indicator does is when the 2 yeast strains mate, they release this compound that turns the indicator blue. So the protein library strain can grow on its own on this SD-3 plate, but we don't want to just pick that up. We want to pick up the colonies that have mated. So we see that the blue colonies are the ones that we have to pick up. We'll pick up those, we'll isolate the yeast DNA, and we'll transform to E. coli, sorry, on an ampicillin plate. Then we isolate the plasmid DNA from that bacteria and send it in for sequencing using the same website that we sent the inserts in for sequencing in the beginning. Then we compare our insert, the confirmed one online, the sequence of DNA Li1, with every single plasmid that we've isolated. And we compare it against all of these, and we see how many times has the insert, has there been a match? How many times has it shown up? And from there, we count how many times it's interacted with each protein. And if it interacts more times, and it's more likely that DNA Li1 binds to that protein for a function rather than a random or non-specific binding. Possible applications of this. Before I joined the lab, like I said, Dr. Zhang had already conducted a yeast-2 hybrid screen, the same technique, but with PACRG, and found that DNA Li1 binds to PACRG for a purpose, which is they form a sperm flagella complex. Blocking this potentially would mean blocking the development of sperm flagella, and like I had said earlier, those are crucial for fertility. So it could lead to the development of male contraceptives. By that line of reasoning, if we find a protein in this yeast-2 hybrid screen that we know the function of, and we block that binding, we can predict what would happen, what it would do to the function, what it would stop. We don't know because we haven't finished the experiment, but we predict that it might have to do with sperm individualization during spermiogenesis, because the multi-tail phenotype that we saw when we knocked out DNA Li1 in mice showed sperm that had not been able to separate and had grown multiple tails. So we think that it could have something to do with that, and we're not sure. It could also serve as a new genomic marker for male infertility, because if we know that these 2 proteins form a complex and that they're crucial to fertility, then we can observe why is this not happening? This will be a new reason or a new explanation for male infertility. With all of that said, we don't know yet, and it was really sad to not be able to finish the experiment before the Discovery to Cure program ended. but I am really, really grateful for the time I did get to spend in the lab and the time I got to spend learning about this project and the other project that Dr. Zhang has been working on, because he was kind enough to let me work on those as well. And with that, I conclude my presentation. I would like to thank Dr. Zhang, Dr. Lee, and Candace for all of their help this summer. Sanjana and I followed them around everywhere, and we never let them take a break from us, but they handled it the whole summer. And also to Dr. Moore and Alyssa for organizing this program, because I had such a great summer. Thank you.
Our next speaker is Sean Jai from Northville High School. Sean?
Hi, my name is Sean J. And in my project, I was finding out whether MNR1 leads to the metabolic flexibility in ovarian cancer cells.
Speak high. Speak high.
So when I was doing my project, my problem was finding out. My problem was patients diagnosed with ovarian cancer have a 30% chance of survival after 5 years. The reason for this is ovarian cancer has an aggressive metastasis, and it goes to metastasis early and is characterized by micrometastasis. Micrometastasis will then lead to occlusion, which then leads to the patient's mortality. This all takes place in the perineal cavity, and the metastasis is easily spread to the GI tract and the omentum. So my general objective was understanding the mechanisms that promote ovarian cancer metastasis. So part of my hypothesis was the metabolic flexibility of ovarian cancer cells is required for the metastatic potential. So based on previous research in the lab, I was able to come up with a more specific hypothesis that MNR1 is a primary regulator for metabolic flexibility of ovarian cancer cells. Metabolic flexibility is when a cancer cell has a stronger mitochondria and is able to survive against inner and outer stresses that normal cells will not usually be able to survive against. So what is MNR1? MNR1 is a protein that regulates genes such as itself and other genes. It also causes anti-apoptosis and improves the mitochondrial respiration or induces ATP production. Depending on the location of the MNR1, the cellular location of it, the proteins it's bound to, it causes the anti-apoptosis or the improvement of the respiration. So my specific objectives. My specific objectives was to first find where MNR1 is located in different ovarian cancer cells. This could be in the nucleus or the mitochondria or even both. And then the second objective was to find what other proteins MNR1 bound to in the different ovarian cancer cells. Does binding to these certain proteins increase the metabolic flexibility of these cancer cells? So the first method, in order to find the first question of where MNR1 is found, I need to use a process called fractionation. And fractionation is a process used to separate the cellular components of the cell. And because the cells are lysed, it separates the proteins that are contained in the nucleus and the proteins that are contained in the cytoplasm, or more specifically, the mitochondria. So in order to do fractionation, I need to do a BCA to get the protein concentration before running the Western blot, and a Western blot to find if MNR1 is found in the nucleus and or the mitochondria of different ovarian cancer cells. The cell lines I used for fractionation was the TKO cell line, the OVCA 432 cell line, R182 cell line, and the F2 cell line. So these are the results. In the TKO cell line, you can see that the MNR1 is located mainly in the cytoplasmic fraction, but I also had to use an integrity control and a loading control when I did the fractionation. An integrity control is to see whether all of the fraction in the cytoplasm is really all cytoplasmic proteins, or did I get some of the cytoplasmic proteins in the nuclear fraction. NDUFS3 is a gene located in the mitochondria, so it should only be in the cytoplasmic fraction of the fractionation. However, there's some contamination, and some of the NDUFS3 appeared in the nuclear fraction of the fractionation. ILF3 is a gene located in the nucleus, and so it should only be present in the nuclear fraction of the fractionation. And as you can see, there's an enrichment of ILF3 in the nuclear portion, and there's a little bit of contamination in the cytoplasmic portion. And GAPDH is used as a loading control to make sure that both proteins were equally concentrated and put in the fractionation. In the F2 cell line, you can see that most of the MNR1 is located in the cytoplasmic portion, and there's a little bit of MNR1 located in the nuclear portion. However, in both the ILF3 and the NDUFS3, there's a contamination of both of them. So based on the conclusion of the first of where it's located in the TKO cell line, majority of the MNR1 is present in the cytoplasmic or the mitochondrial fraction of the fractionation. And the F2 cell line, I cannot conclude whether MNR1 is in the cytoplasmic or the nuclear fraction because of the contamination located in the nuclear and the cytoplasmic fraction. So in order to find what proteins MNR1 is bound to, I used a process called immunoprecipitation. Immunoprecipitation is where there's a set of beads, and I put the lysate on the set of beads, and the non-specific proteins bind to the set of beads, and I take out the lysate, and the lysate has now specific proteins, and when taken out, I labeled that the supernatant. The supernatant has all the proteins that I'm looking for, and all the non-specific proteins that bound to the bead were thrown away. And then next, a new set of beads were put, and an antibody for the proteins I'm looking for was put in the set of beads. And the lysate was also put on, and the beads bound to the antibodies, which bound to the proteins I was looking for. The lysate, which now didn't contain the proteins that I was looking for, was taken out and labeled the wash. And the set of beads now contain the antibody and the proteins I was looking for. And then once the set of beads were taken out, I was left with the protein I was looking for. The reason I use this is because MNR1, if it was bound to the proteins I'm looking for, which I'll explain later, which are COX4 and BCLXL, when Western blotting them, I would be able to see both the BCLXL and the MNR1 because they would be bound together, or the COX4 and the MNR1 because they're bound together. So in order to do this, I lysed the proteins, which broke down the cells, and then captured proteins for analyzation. Then I did a BCA to get the protein concentration, and then I immunoprecipitated to isolate MNR1 and what is bound to in different ovarian cancer cells, and then I Western blotted it. However, when isolating the MNR1, because there is no antibody for it, I really isolated the BCLXL and the COX4 first to see if it was bound to the MNR1. So what is BCLXL and COX4? So when MNR1 is bound to BCLXL, which is another protein, it causes anti-apoptosis. And when MNR1 is bound to COX4, it induces cellular respiration. So the cell lines I used for this were the TKO cell line, the MR182 cell line, and the F2 cell line. So this is the immunoprecipitation, and as you can see, the IgG is used as a control, and then the COX4 is the data I'm looking for. So as you can see, there's COX4 present in the input, which shows that there's COX4 in the F2 cell line, and then there's also COX4 in the IP. However, in the IP, there's no MNR1, showing that when I found the COX4, there was no MNR1 bound to it. So this is the BCLXL IP. As you can see, there's BCLXL in the input, and there's BCLXL in the IP. However, there's no MNR1 in the IP, meaning there's no BCLXL and MNR1 bound together. In the TKO cell line, you can see that there's COX4 in the input, and there's also a little bit of COX4 in the IP. However, there's once again no MNR1 in the IP. But for BCLXL, there's a lot of BCLXL in the IP, and there's some MNR1 in the IP also, showing that when I immunoprecipitated for BCLXL, MNR1, because it was bound to the BCLXL, came with it, and in the Western blot, both were present. So what I can conclude from this is in the TKO cell line, MNR1 is shown to be bound to BCLXL, and the F2 cell line, the results are inconclusive. So in my general conclusion, in TKO, most of the MNR1 is found in the mitochondria, and most of the MNR1 bound is shown to be bound to BCL-XL. In F2, MNR1 is present in both fractions, but it's not clear where a majority is located because of the contamination. And because of this, the results could not demonstrate whether it binds to COX-4 or BCL-XL. And that's the end of my presentation. I would like to thank Hussein and Dr. Alvaro for helping me out in my project, and Alyssa for doing all the organizing it all. Thank you.
Any question? No questions? No suggestions? Very good, my friend. Thank you.
Thank you.
Our next presenter is Sanjana Kandiraju from International Academy Central.
Hi, everyone. My name is Sanjana Kandiraju, and I'm a senior at International Academy Central. And over the past 8 weeks, I've been working in Dr. Zong's laboratory, and my project is on the role of IFT 172 in Leydig cells. So the overall objective of my project is to generate IFT 172 knockout Leydig cells in order to explore its effect on the cell line itself, hormone production, and germ cell development. So before I go into my project, I would like to give a little bit of background information on some of the terms I will be using. So what exactly are Leydig cells? Leydig cells are somatic cells located in the male testes, and I will specifically be using the MA10 Leydig cell line. And the main function of Leydig cells are to produce sex hormones, such as testosterone, necessary for sperm formation or spermatogenesis. And on this slide, you can see a picture of Leydig cells our lab took. Now I'll be talking a little bit about what cilia is. Cilia are hair-like organelles located all over the surface of a cell, and they're similar to the organelle called the flagella. And cilia has two main functions. Number one is that their movement and vibration is able to give cells motility. And number two, it is also used to transport cellular materials. And if there are any abnormalities in cilia, this can give rise to an array of disorders that are characterized as ciliopathies. So the image on this slide shows a diagram of a cilium, and on this diagram, you can see the IFT raft. So the purpose of the IFT raft is to transport proteins down the microtubules to the cilia in order for cilia to be formed and to maintain cilia itself. So that's the connection between IFT and cilia. Next, what exactly is IFT? So IFT is the intraflagellar transport complex, and there are 22 IFT components that have been discovered, including IFT 172. And the IFT complex is highly expressed in ciliated cells, particularly in the testes, as well as in the trachea and in the brain. And the IFT complex is essential for cilia formation, as we saw with the IFT raft, as well as male germ cell development. And specifically IFT 172, it's a gene that translates for two major proteins. One is a full-length 172 kDa protein, and the other is a smaller 130 kDa protein. And both are highly expressed in the testes and in somatic cells, including the Leydig cells. So in the Western blot at the top, you can see two strong protein bands at the 170 kDa and 130 kDa markers, and this is under the testes column. And then in the Western blot on the bottom, you can see a similar pattern for a Leydig cell line. So overall, this suggests that IFT 172 proteins are highly expressed in Leydig cells and in cells in the testes. So how exactly are we going to be knocking out the IFT 172 gene in mammalian cells? So we will be using the technique called the CRISPR-Cas9 technique. And the first step in this process is to create a target sequence for IFT 172 through a website called Thermo Fisher. So IFT 172 is a long sequence of nucleic acids, and for the CRISPR-Cas9 technique, you can only use a short sequence. So that's why we create a target sequence. Next, the company will send us back the single-stranded forward and reverse oligonucleotides of our target sequence, and we will have to anneal them or bind them together to create double-stranded DNA. Our next step is to clone the double-stranded DNA, insert into the GeneArt CRISPR nuclease vector provided by the GeneArt CRISPR kit. So we are inserting our double-stranded DNA into this vector, and it'll fit right where the arrow points to in between those overhangs, sort of like a Lego piece. And then on our vector is the OFP, or the orange fluorescent protein. And this will help us later on to identify our positive cells, because if a cell has the plasmid in it, it'll create these proteins that fluoresce an orange color. So next, how exactly is IFT going to be knocked out using this technique? So a guide RNA will bind to our IFT 172 target sequence, and this can be at many locations on the IFT 172 gene, which is why we can have multiple target sequences. So all a guide RNA needs is an NGG, which is a three nucleic acid sequence that contains any nucleic acid followed by a guanine and another guanine. And then this complex acts as a tag for Cas9, and Cas9 will find this complex and it'll cut IFT 172, which will inactivate the gene, and it can no longer produce its proteins. So in the image on this slide, you can see that there's a DNA sequence which represents IFT 172, and then the yellow represents the guide RNA that will bind to its complementary IFT 172 sequence, and then the red structure is Cas9, and it'll cut the gene. So after we have inserted the knockdown of IFT 172 into the GeneArt CRISPR plasmid, we have now created a vector, we have now created a plasmid. So the next step is to transform our ligated plasmids with IFT 172 knockdown into bacteria. Then we will grow the bacteria colonies on bacteria colonies on an ampicillin-resistant LB agar plate, which is an antibiotic. And then we will pick up 8 white bacteria colonies and grow them further in LB broth with ampicillin. Next, we will isolate the plasmid DNA, and then we will sequence the plasmids through a website called GeneWiz to confirm that our plasmids have the correct IFT 172 sequence. So the 3 agar plates that you see are 3 different locations at which the CRISPR-Cas9 technique took place, and the image on the bottom shows our sequence. So the nucleic acid sequence on the top is our target sequence that we created, and the sequence on the bottom is what we received from GeneWiz. And as you can see, they are 100% match, which means that we can move forward with the next part of our process. So after we have confirmed that our plasmids have the correct insert, we can now transvect the plasmids into Leydig cells. So we will grow the MA10 Leydig cells, and then we will transvect them. And in order to transvect them, we have to combine a few things. So the first is DMEM-F12 containing 10% FBS, which is essentially a cell culture medium. And then next, we will add in a transvection reagent, which will help the plasmid enter the cell by making it more lipid soluble. So the plasmid is foreign DNA, so it needs a little bit of help in order to enter the cell. And then we will also mix in the plasmid DNA itself, and we will allocate this mixture to a 6-well plate that contains MA10 cells. So the images on this slide show the transvected MA10 Leydig cells that were grown in DMEM-F12, 10% FBS, 0% horse serum, and 5% carbon dioxide. However, the images show that these cells did not grow as well as they should have grown. So on the top, you can see that there is a red cell, but it's not because of the orange fluorescent proteins, but instead it's because of the autofluorescent dead cells. And the image on the bottom more clearly shows how a lot of these cells died. So any circular cells you see are examples of dead cells. So with that, we decided to change our cell culturing conditions. So we have now changed the conditions in which we grow the MA10 cells in before transvection. So now we are growing them in DMEM-F12, 5% FBS, 2.5% horse serum, and 3.5% carbon dioxide. And after changing these cell culturing conditions, the MA10 cells are growing much better. So in the image, you can see that there are a lot more live cells as well as a lot more positive cells. So 2 of the cells that I've circled are examples of cells that are fluorescing a red color, which could mean that the plasmid successfully entered the Leydig cells and are now producing those orange fluorescent proteins. This could also mean that they are autofluorescent dead cells, but overall, after changing the conditions, our cells are growing a lot better. So our conclusions is that we have successfully created plasmids that have IFT 172 knocked out in them, and we have also optimized the conditions for cell culturing the MA10 Leydig cells. So in the future, we will transvect more newly grown MA10 cells with our plasmids, and then we will sort the positive cells using the orange fluorescent protein to generate a stable cell line. And after we have generated a stable cell line, we will then test the positive IFT 172 knockout cells using specific antibodies in a Western blot to analyze proteins. And ideally, we shouldn't see any IFT proteins because the gene was knocked out. And after we test this, we can then analyze sex hormone levels such as testosterone to see if it's affected by the knockdown of IFT 172. So we will measure both cell medium and lysate under basal stimulation and conditions. And if there is a change in sex hormone levels, we know that one of the major functions of Leydig cells is affected by the knockdown of IFT 172, and that can in turn affect sperm formation and male fertility. Lastly, we will generate Leydig cell-specific IFT 172 knockout mice to analyze in vivo function. So that is it for my presentation. Thank you all so much for listening. And I would like to give a big thanks to Dr. Zong, who is my PI, Candace, Dr. Lee, and everyone in the Discovery to Cure program. You have all been such vital parts in my end product here. And now I will leave room for any questions. Thank you.
Do you have any questions for her? Any questions for you guys? I don't want to put you in the spot. You have good friends. Very nice.
Thank you.
Thanks.
Our next presenter is Laura Stefan from International Academy East.
Starting again this time. I'm Laura Stefan, and my project this summer was characterizing the immunological inflammatory response after a cell is infected with both MHV68 and LPS. So just some back information about pregnancy. During pregnancy, there are a number of immunological and physiological changes that occur to support the fetus. The placenta here is of important nature. As it is derived by the fetus, it provides essential nutrients and oxygen to it and removes waste products from it. The maternal side of the placenta is called a decidua. It protects the fetus from a maternal immune rejection as well as infections. And on the right shown here is the maternal fetal interface. There are a lot of immune cells such as T cells and macrophages, and it is also mainly composed of placental or trophoblast cells as well as decidual stromal cells. So viral infection, after someone is exposed to a viral infection, a subsequent bacterial infection will happen, and then inflammatory response will be produced. Inflammation is when a lot of immune cells are being recruited to the site of infection, producing pro-inflammatory cytokines to exacerbate the immune response. During viral infection, it's characterized when yeah, the camera. It's okay. It's okay. It's okay. During viral infection is when a virus is successfully reproducing inside of its host, causing a sequential inflammatory response. Now, during pregnancy, excessive inflammation can be dangerous to both fetus and mother. For example, it can cause preterm labor, preeclampsia, and for the child, it could cause neurodevelopmental issues, and the figure here displays such. So the adverse effects I just mentioned were manifested in the Spanish flu pandemic of 1918. Pregnant women, when exposed to a viral infection, were more susceptible to bacterial infection than normal pregnant women. Mortality rates amongst pregnant women with bacterial infection were abnormally high. And then also developmental issues for the fetus were manifested. However, there was no viral RNA detected on the fetal side, which suggests that the inflammation was crossing the placental border and then infecting the fetus. So in my project, I'll be characterizing this inflammatory response in hopes that future research could create a drug that would inhibit these detrimental excessive inflammation effects. So to do this, we use two entities, MHV68 and LPS. MHV68 stands for murine gamma herpes virus 68. It is a naturally occurring rodent pathogen, and in my project, it will be acting as a bacterial infection for our cells. A common property of this virus is to modulate the immune response, which is also present in our experiment results. LPS, also known as lipopolysaccharides or endotoxins, are composed of O antigen and gram-negative bacteria, and LPS is known to induce an immune response. So collectively, these two entities can effectively model what happens in a bacterial infection and subsequent inflammatory response. So type 1 interferons, oh my bad, let's skip this slide. Oh, wait. No, this is going forwards.
Use the.
Okay, there. So LPS best responds to TLR4. After LPS binds to TLR4, it can induce two downstream pathways, one of which is the MYD88 dependent pathway, which would then induce inflammation such as TNF alpha, IL-6, IL-12 alpha. And the other side is the MYD88 independent pathway, which would then induce type 1 interferons. So type 1 interferons are key antiviral interferons. They would then induce ISGs or interferon signaling genes that would then produce pro-inflammatory proteins that would initiate the innate immune response. So our hypothesis, MHV infection during pregnancy leads to susceptibility to bacterial infection and a subsequent inflammatory response. So in order to prove our hypothesis, we had two cell lines, our trophoblasts or swan cells, as well as our decidual stromal cells, also known as HESC cells. Our four sample types are as follows: wild type plus saline, wild type plus LPS, MHV68 plus saline, and MHV68 plus LPS. In order to come to a conclusion, we use a lot of qPCR to have our results. Our main aim: does MHV68 modulate the LPS-induced inflammatory response at the maternal-fetal interface? So we will first be looking at toll-like receptors, or TLRs. So looking at our decidual cells first, we see that for TLR3, there is a decrease in the regulation for this molecule comparing wild type plus LPS and MHV68 plus LPS, whereas for TLR4, we see a relatively no change in the expression of this molecule. For our trophoblast cells, we see that there is a decrease in the expression of TLR3 comparing wild type plus LPS and MHV68 plus LPS, and the same results follow for TLR4. So what does this tell us? In trophoblast cells, we see the downregulation and transcription of TLR3 and 4, and in the decidual cells, we see there's no change in its regulation of TLR4. This suggests that MHV68 is preferentially modulating the immune response in these decidual cells. So we will now be looking at interferons. So firstly, for the decidual cells, we see that for interferon alpha, there's an increase in the expression for this molecule. However, for interferon beta, we see a decrease in the expression comparing wild type plus LPS and MHV68 plus LPS. The data for interferon beta suggests that MHV68 is indeed modulating the immune response. Now, from this, we can conclude that it is not modulating all type 1 interferons, rather just interferon beta. Looking at interferon lambda, a type 3 interferon, we also see that modulation occurring, which makes us hypothesize that is also modulated through interferon lambda. And then for our trophoblast cells, we see that for interferon alpha, there's a relatively no change. And for interferon beta, we further see the modulation occurring, and no results were detected for interferon lambda in our swan cells due to a high CQ value in our qPCR. So in summary, MHV68 suppresses in both cell types. However, MHV68 differentially modulates interferon alpha. We will now be looking at ISGs. So for our decidual cells, for MIX1, as we know, this is a molecule that is induced by interferon beta. So seeing the decrease in expression for this entity, we can validate our results for interferon beta. We do indeed see the decrease comparing wild type plus LPS and MHB68 plus LPS. For ISG15 and ISG20, we see either a no change or an increase. And knowing that these two types of ISGs can stem from type 1 interferons is not something that is surprising. And then for our SWAN cells, for MIX1, we can further validate our data from interferon beta, knowing that there is a decrease in expression for MIX1. And for ISG15 and ISG20, we also see the decrease in expression for these molecules. So takeaway, the effect of the suppression of interferon beta is manifested with the ISGs that interferons induce in both cell types. In other words, with a decrease in expression of interferon beta, it leads to a decrease in expression of MIX1. And now we'll be looking at inflammation. So for our decidual cells first, we see an increase in inflammation now, comparing wild type plus LPS and MHB68 plus LPS. For IL-17 especially, we do see great increase in expression for these entities. The same thing follows through for IL-12 alpha, IL-6, and TNF alpha. For the SWAN cells, we see a decrease in induction of inflammation looking at TNF alpha, IL-6 especially, comparing wild type plus LPS and MHB68 plus LPS. And for IL-12 alpha and IL-17, we see a no change. So for going back to the decidual cells, we can hypothesize that the suppression of the MYD88 independent pathway leads to the hyperactivation of the MYD88 dependent pathway, inducing this inflammatory response. Inflammation will then only occur in the decidual stromal cells and not in the trophoblast cells. Our overall conclusion, MHB68 is preferentially modulating the immune response, and further, the inflammation stemming from the decidua and not the placenta. And I would take a few moments for acknowledgements. I would like to thank Anthony for your time and patience. I couldn't have done this with a lot of your help. And Dr. Moore, I'd like to thank you for this opportunity. It was unlike any other. And I also want to thank a lot of the faculty members, Dr. Alvaro, Dr. Ding, Dr. Tisha, Anna, Annie, Sandra, Yuan, Hussein, and Alex for all of your help in guiding me throughout my project this summer. Thank you. Any questions? No questions?
Okay, very good. Thank
you.
Our next presenter is Claire Wu from Troy High School and Madison Parnello from Regina High School.
Hi, good morning, everyone. My name is Madison Parnello, and I'm from Regina High School. My name is Claire Wu, and I'm from Troy High School. And today our presentation is titled, Baby, You're Stressing Me Out. And we know the title is a little cheesy, but today we're going to be talking about babies and stress. More specifically, we'll be talking about how pregnancy-related stresses affect the uterus, especially on a molecular level. All right, so Madison and I are in Dr. Condon's lab, and since our results are closely related, today we're doing a joint presentation. So our lab works on defining the mechanisms that result in preterm birth. And this research is really important because here in the US, we have the 6th highest rate of preterm birth on the planet, with 1 in every 10 babies being born preterm.
At a local level, these rates of preterm birth are even higher. Detroit in particular has a preterm birth rate of 18% within the Black population. Similar rates exist in Pakistan, Zimbabwe, and Malawi.
Okay. So during pregnancy, every uterus experiences several stresses, and these stresses can be physiological, chemical, or even hormonal. So examples of these stresses are hypoxia, mechanical stretch, hypertrophy, inflammation, cytokine fluctuation, and hormone fluctuation. So to elaborate on these stresses, hypoxia is low oxygen levels. Mechanical stretch occurs because the uterus has to stretch in order to accommodate the growing fetus. Hypertrophy is increased cell size, and it's the predominant method of uterus growth during pregnancy. It occurs as a result of mechanical stretch. Inflammation is an immune system response, and it's the body's reaction to the fetus implanting. Inflammation is also needed for parturition. Cytokine fluctuation occurs because cytokines are mediators of the inflammatory response. And as we just talked about, inflammation is necessary during gestation. Lastly, we have hormone fluctuation. And this is because hormones like estrogen and progesterone fluctuate throughout the duration of a pregnancy. So in the pregnant uterus, normal pregnancy-associated stresses activate the unfolded protein response, which prevents the onset of premature uterine contractions and therefore preterm birth. So we'll talk more about the unfolded protein response in the upcoming slides. So the endoplasmic reticulum is the site of the unfolded protein response, and the unfolded protein response, the endoplasmic reticulum exists within almost every single cell of the body. It has several functions, such as within protein synthesis, protein folding, lipid synthesis, calcium storage, and detoxification. Our lab is most interested in the endoplasmic reticulum's role in protein synthesis and folding. So when protein synthesis or folding is impaired, there's an accumulation of misfolded or unfolded proteins, and this leads to endoplasmic reticulum stress. Since the cell's goal is always to maintain endoplasmic reticulum homeostasis, this stress must be resolved. And the cell does this by triggering the unfolded protein response. So the unfolded protein response has several constituents, the first of which is expanding the endoplasmic reticulum by increasing lipid synthesis. And this works because the ER is composed of lipids. So the second option for the cell is to attenuate protein synthesis, which reduces protein load. Thirdly, the cell can remove misfolded proteins through ER-associated degradation. And the cell needs to do this because misfolded proteins are non-functional. And lastly, the cell can produce chaperones and protein processing enzymes. Chaperones help protein folding, and both of these are essentially expanding the machinery to fold proteins. So there are three main pathways that ER stress triggers and that constitute the unfolded protein response. So each of these pathways starts with the transmembrane protein that's embedded within the endoplasmic reticulum. And these three pathways are IRE1, ATF6, and PERK. So for the IRE1 pathway, XBP1 becomes spliced. And this spliced version of XBP1 goes on to act as a transcription factor and is able to upregulate the production of chaperones, increase lipid synthesis, and also trigger ER-associated degradation. So once again, ER-associated degradation removes misfolded proteins from the cells. The second pathway is the ATF6 pathway, and ATF6 becomes cleaved. This cleaved version is able to upregulate genes coding for ER chaperones and ER-associated degradation components. Lastly, we have the PERK pathway, and it results in the phosphorylation of EIF2 alpha. The phosphorylated version of EIF2 alpha is able to cause general inhibition of protein translation. CHOP upregulation also results from the PERK pathway, and CHOP induces apoptosis or cell death by eventually activating caspases. So in most cells, CHOP causes apoptosis. However, in pregnant uterine cells, non-apoptotic caspase 3 becomes activated, which helps maintain uterine quiescence. And uterine quiescence just means that the uterus is not contracting before it's meant to. All right, so the three pathways that we just discussed are typically kept inactive by the three transmembrane proteins binding to BIP, and BIP is a chaperone protein. So when the cell encounters ER stress, BIP is able to dissociate from the three transmembrane proteins and instead bind to the misfolded proteins. So this has two functions. First of all, it activates the three pathways to help reestablish ER homeostasis by triggering the unfolded protein response. And secondly, it prevents the misfolded proteins from aggregating, which causes harm to the cell and to the body. Okay. So these types of stresses that we just previously discussed can either be local or systemic. So we wanted to identify if uterine quiescence and the unfolded protein response are regulated locally or systemically. So if they're regulated systemically, things like cytokine fluctuation and hormone fluctuation would be controlling these processes. On the other hand, if they were regulated locally, things like hypertrophy or mechanical stretch would be controlling these processes.
All right, so these types of stresses that were just previously discussed can either be local or systemic. You can see in the picture to the left, the effect on the fetuses of the effect of the fetus on the mom's pregnant uterus would be considered a local type of stress. Whereas on the picture to the right, pregnancy-associated circulating factors affecting the mom's uterus would be considered a systemic stress. Therefore, it would be something involving the blood of the pregnant mother. And we have determined which uterine proteins are affected in a systemic or local manner during pregnancy in the next few slides. So the next graph is changes in estrogen or progesterone. An example of systemic factors would be these two hormones. These are different types of chief hormones fluctuating throughout a rat uterus during pregnancy in the graph. Now, looking at the dotted line, you can see that progesterone decreases towards term, while estrogen increases towards term. These hormones are vital considering that estrogen enables the uterus and placenta to move, causing vascularization, which means it helps with the formation of blood vessels. While progesterone plays a role in preventing the uterus from contracting early. So therefore, these would be systemic factors. So here's the methods portion of our presentation. All right, so our lab, we use the unilaterally pregnant rat model to examine the effects of these local and systemic factors. This model was generated by taking a non-pregnant rat, which is a rat with a biocarnate uterus, meaning it has two uterine horns. We tied off one horn just below the ovary so that when mated, the eggs from the tied off uterine horn will never get fertilized. And you can see in the pictures to the left, the little string. So once we tied those off, they would never get fertilized, while the other unligated horn does get fertilized. These tissues were then isolated from the empty unfertilized horn and the gravid fertilized horn and cleared of all fetal material. Cytoplasmic and nuclear proteins were extracted and probed for the proteins of interest by a Western blot analysis, and these samples were collected from gestation day 6 to gestation day 23, which is term for pregnant rats. We then examined these tissues for markers of the unfolded protein response and inflammation and prostaglandin synthesis, all crucial components that regulate the onset of uterine contractions at term. So in this model, both the empty and gravid uterine horns will be subjected to the circulating systemic changes associated with pregnancy, but only the gravid uterine horn will experience the local fetal driven consequences. And to the right, you can see an example of a human uterus. So here's the Western blot protocol that we followed for our experiments. So first, we would run a gel by loading a sample into each well. And once the gel is running, the proteins separate based on size. And you can see to the left is the gel running, on the bottom is all the proteins, and then to the left is called a ladder. So next we perform a transferring process in which the proteins in the gel are transferred onto a membrane, and the only visible colors on the membrane is the ladder part, in which serves as a marker for the protein, so we know where to cut. So as shown to the right, you can see the membrane. So after the transferring is done, we block the membrane using a milk buffer to reduce antibody and background proteins on the film. Once the membrane is blocked, we apply primary antibody, and that will only bind to our protein of interest. Next, we apply secondary antibody, and this binds to our primary antibody. And the final step is to apply chemiluminescence, in which will bind to the secondary. So lastly, we pick up the signal from the chemiluminescence by placing a film onto the membrane, developing it, and you can see our film results in the next.
So this is the Kumasi blue stain gel. So it just shows the proteins that are remaining on the gel. Okay. So first of all, the goal of a Western blot is to examine differences in expression of a certain protein across different tissue samples. And these tissue samples may be treated differently or be from different time points. Essentially, they show some sort of manipulation in the independent variable that you want to test in the experiment. So loading controls are proteins that have high consistent expression across all the samples being studied. And loading controls have a few roles. First of all, they quantify the amount of protein loaded in each well. And secondly, they check if proteins were evenly transferred from the gel to the membrane. So essentially, they normalize unintended variation, such as due to differences in loading between the different samples. So on the bottom of this slide is a Western blot for PDI. And the Western blot was performed on samples prepared from uterine tissue from the unilaterally pregnant rat model. Tissue was collected from day 6 to 23 of gestation, both from the empty, unfertilized, and gravid fertilized uterine horns. And PDI proved to be a good loading control, as evidenced by its relatively consistent expression from day 6 to day 23 of gestation. There's also similar levels between the empty and the gravid uterine horns. All right, so these proteins on this slide are not suitable to be used as loading controls. So we have tubulin, calponin, SRC3, and GAPDH. And they're not suitable to be used because you can see that protein expression changes from day 6 of gestation to postpartum. We also see differences between the empty and the gravid uterine horns. So interestingly, in other species, these proteins have proved to be good loading controls. So we can see that there's a lot of differences between a rat pregnancy and a human pregnancy, for example.
All right, so next is the results part of our presentation. So this diagram portrays how prostaglandins, a type of lipid compound, are made. So when prostaglandins are produced, the uterine compartment, they promote uterine contractions. You can see that caspase-3 activation, which is written in red here, leads to phospholipase A2 cleavage, generating arachidonic acid. The conversion of arachidonic acid to prostaglandin depends in part of NF-kappa B activation, as you can see in the diagram to the right. So we know that the unfolded protein response prevents prostaglandin production earlier in pregnancy by maintaining caspase-3 in a non-apoptotic status. And we also know that the unfolded protein response can limit inflammation, again, acting to limit prostaglandin production. So therefore, the woman does not go into labor. So increase of prostaglandin leads to increase of contractions. Therefore, we looked at the components of the unfolded protein response, BIP and CHOP, to see how these proteins were regulated. Okay, so on the graph, you can see BIP and CHOP here. So from early gestation, BIP upregulation only occurs in the gravid uterine horn, suggesting its upregulation is dependent upon the presence of the fetus, whereas in the empty uterine horn, it remains unchanged. So in contrast, CHOP levels in early gestation are very high in both the empty and gravid uterine horns, suggesting that CHOP activation is regulated in a systemic manner. However, from gestation day 17, we see a sharp decline in CHOP in the gravid uterine horn, with its decline to undetectable changes in both uterine horns as term approaches. This data demonstrates that BIP is likely regulated by the presence of the fetus, whereas CHOP is likely regulated by circulating factors. So also in the graph, you can see the light blue bar is our empty, while the dark blue bar is our gravid. So BIP is a cytoplasmic protein. So PDI was used as our loading control, and CHOP is a nuclear protein. So pulling PARP was utilized as our loading control. And we utilize these as our loading controls because their expression remains pretty stable across gestation. So the next protein is caspase-3, and you can see at gestation day 16, they are regulated equally in both the gravid and uterine horns. However, throughout gestation to term, the gravid uterine horn is highly active for non-apoptotic caspase-3 activation. So as discussed earlier, caspase-3 activation is normally associated with apoptotic cell death. However, in this model, cleaved PARP is isolated to the empty uterine horn and never appears in highly caspase-3 positive gravid uterine horns, as measured by PARP cleavage on the second plot. This indicates that caspase-3 in the gravid uterine horns is non-apoptotic.
Yeah.
So in the section, along with the increase in caspase-3 levels to term, we also observe IPLA2, which is a hallmark for increased prostaglandin production, as I mentioned earlier, which occurs solely in the gravid uterine horns. So overall, this demonstrates that caspase-3 activation and downstream prostaglandin production are both dependent on the presence of the fetus and not regulated by circulating factors. Therefore, they are considered a local factor. And you can see below, we have PDI as our loading control, and then above is the protein IPLA2.
Okay, so the second section of our results has to do with the role of inflammation throughout gestation. So to begin the pathway seen on the slide, we have toll-like receptor 4. Toll-like receptor 4 goes on to activate phospho-TBK1, and phospho-TBK1 activates transcription factor NF-kappa B. NF-kappa B causes inflammation, so you can see how this is an inflammatory pathway. And NF-kappa B also increases prostaglandin production. Since prostaglandins are what trigger uterine contractions, NF-kappa B helps regulate the duration of pregnancy. So the unfolded protein response that we discussed earlier actually inhibits NF-kappa B's role in prostaglandin production, which holds off labor. So essentially, during the earlier stages of pregnancy and throughout the most of pregnancy, the unfolded protein response triggered by gestational stresses prevents premature labor. So we mainly focused on the first three proteins seen in this pathway: toll-like receptor 4, phospho-TBK1, and NF-kappa B. All right, so first we have NF-kappa B, and as we saw in the previous slide, it mediates gestation by causing inflammation and also activating prostaglandin synthesis. NF-kappa B targets inflammation directly by increasing production of cytokines, which help mediate the inflammatory response. So with the Western blot of NF-kappa B, which you can see on top, we normalize the data with loading control PARP. Specifically, we used full-length PARP, which you can see the arrow pointing to. We then put the normalized data into a graph with the empty uterine horn being indicated with light blue and the gravid uterine horn being indicated by dark blue. So we can see how NF-kappa B is upregulated in the gravid uterine horn every day after day 6 of gestation. So this indicates that the presence of the fetus somehow causes a local increase in expression of NF-kappa B. And if we take a look at day 23 specifically, we can see how NF-kappa B is especially elevated in the gravid uterine horn. And this is because inflammation, which NF-kappa B leads to, is essential for parturition or giving birth. So next we have TLR4 and phospho-TBK1, which are upstream of NF-kappa B and therefore also help mediate inflammation and prostaglandin synthesis. So we normalize the data from the Western blots of TLR4 and phospho-TBK1 with loading control PDI. We then put this normalized data into graphs with, once again, the empty uterine horns being indicated by light blue and the gravid uterine horns being indicated by dark blue. So toll-like receptor 4 in other species has been described to be upregulated at the end of pregnancy to allow for increased inflammatory activation. And we observed something really similar here in the pregnant rat. So in the pregnant rat, TLR4 levels increase towards term in a gravid horn specific manner, showing that the presence of the fetus causes a local increase in expression of TLR4. TLR4 and phospho-TBK1 show similar patterns of variation across gestation, due to phospho-TBK1 being directly downstream of toll-like receptor 4. And you can see the similar pattern in both the Western blots and also the graphs. So they all have relatively high expression in early gestation, lower expression in the middle of gestation, and are again upregulated towards the end of gestation, especially in the gravid uterine horns. So in early gestation, both TLR4 and phospho-TBK1 show high levels of expression because of the inflammation that implantation of the fetus causes. If we take a look at phospho-TBK1 specifically, we can see a really elevated protein expression in the gravid horn of day 23. So this indicates that the bulk of inflammation occurs right as the rat gives birth. All right, so moving on to our conclusion slide. So our analysis demonstrates that many of the proteins in the prostaglandin pathway, such as TLR4, IPLA2, and phospho-TBK1, are all regulated locally by the presence of the fetus. So the presence of the fetus through things like mechanical stretch or hypertrophy causes local responses in late gestation, especially in days 21 and 23 of embryonic development. So if you look to the bottom right of the slide, you can see TLR4, IPLA2, phospho-TBK1, and you can also see day 21 and 23 of gestation being indicated. So the unfolded protein response serves to inhibit the production of prostaglandins, and the proteins involved in the unfolded protein response appear to be regulated in many different ways. BIP is one of these proteins, and it seems to be locally regulated consistently throughout gestation, with the gravid uterine horn always showing higher levels of expression than the empty uterine horn.
So on the other hand, CHOP appears to be systemically regulated for the majority of gestation. CHOP is elevated in both uterine horns early in gestation and declines to undetectable levels in both uterine horns at term. Circulating hormones such as estrogen and progesterone, which changes term approaches or circulating inflammatory cytokines, could be the mechanisms behind CHOP regulation. So this analysis increases our understanding of local versus systemic changes that occur during pregnancy and reveal the dynamic interplay between systemic and local changes that will likely work together to promote an appropriate gestational length and the prevention of premature birth.
All right, so next we have some future research possibilities. So we know that CHOP is regulated in a systemic manner, potentially by hormones like estrogen and progesterone. So since progesterone typically decreases towards the end of gestation, a possible research idea is to keep progesterone levels high throughout pregnancy and examine the effects that has on CHOP expression. Similarly, we could keep estrogen levels constant throughout gestation to examine the effects estrogen levels have on CHOP expression. So a second, more general research idea is we can further explore the factors that affect NF-kappa B expression across gestation. And this is because both TLR4 and phospho-TBK1 are directly upstream of NF-kappa B, yet show a slightly different pattern of variation across gestation. So research could be done in this aspect.
All right, we'd like to thank Dr. Condon, Dr. Surya, Francis, and Wayne State University and the Discovery to Cure program for giving us the opportunity to be able to present and learn in our research.
Last but not the least, Layla Johnson from Northville High School and Vidya Harini Srinivas from Troy High School.
Well, thank you, Maddie and Claire, for that wonderful presentation. My name is Layla Johnson, and I'm an incoming senior at Northville High School.
My name is Vidya Harini Srinivas, and I'm an incoming senior at Troy High School.
So during our 8-week-long internship, we had an amazing time working under Dr. Surya studying preterm birth. Although we work closely with Dr. Condon and her mentees, Harini and I have centered our focus in studying the epitranscriptomics behind uterine quiescence, focusing mainly on mice and human pregnancies. Our presentation is called Methylation in What Station? Gestation, and we hope you enjoy it. Enjoy our baby.
Oh, there's a baby.
So to begin, I would like to discuss the fetal stages of mice. Unlike human pregnancies, which usually last between 37 to 42 weeks of gestation, mice pregnancies are much shorter, and they only last 19 days. Although the length of gestation differs between mice and humans, the general idea of stretch in both pregnancies are the same. So in this picture, it shows a period of 10 days of growth in a mouse fetus. The greatest periods of growth occur between day 11 and day 13. And then if you look, day 17 and 19 are probably the least growth occurrence, because this is when like the final stretching of the uterus occurs. And this stretching is due to ER stress and the unfolded protein response.
Next, we have ER stress during gestation. Since the previous group spent a lot of time covering ER stress and the unfolded protein response, we will focus more on one component of response: translation and the phosphorylation of PERK. When GRP78, also known as BIP, dissociates from PERK under ER stress, it activates the phosphorylation of PERK. This activated PERK then phosphorylates the eukaryotic initiation factor 2-alpha, leading to a general inhibition in protein translation. However, the phosphorylated EIF2-alpha also preferentially increases the translation of the transcription factor ATF4, which in turn upregulates CHOP. Failure to lessen ER stress then leads to CHOP-mediated apoptosis, also known as cell death. This process is evidenced by the Western blot you see on the slide. As we see an increase, but then a decrease in BIP expression during the gestation period, there's a decrease in CHOP expression, but an increase in phosphorylated EIF2-alpha and phosphorylated PERK expression. Next, let's take a look at CAP-dependent and CAP-independent translation. CAP-dependent translation begins with the eukaryotic factor 2 binding to the 43S ribonuclear protein complex. Then the EIF243S complex binds to the 4E, 4G, and 4A at the 5' cap, initiating translation in a CAP-dependent manner. Lately, it has been found that in CAP-independent, also known as M6A-mediated translation, capped mRNAs don't need the EIF2 complex as they are translated during times of ER stress. The M6A mark in the 5' UTR works as a substitute for the 5' cap by recruiting the 40S complex, ultimately triggering CAP-independent translation.
Now we're going to center the discussion around M6A and its various functions. From this diagram, you can see that M6A not only plays a role in CAP-independent translation, but also transcription, alternate splicing, alternate polyadenylation, nuclear export, mRNA degradation, and mRNA stabilization. In this presentation, we will mainly be focusing on alternative splicing and translation. Let's first take a look at alternative splicing and M6A's role in it.
Very simply, when we talk about alternative splicing, we are referring to splicing a gene at its splice sites. In general, a gene is made up of both introns and exons. Alternative splicing is a process during gene expression in which certain intron sequences are moved. The final mRNA thus consists of the remaining sequences called exons, which are connected to one another during the splicing process. Now, you may wonder how this is related to HNRNPG, which is shown on the slide. Well, recently there was a paper, which is named on the slide, that discusses M6A's involvement in alternative splicing. Specifically, the paper talks about how HNR&PG binding of mRNA is enhanced by M6A marks and how HNR&PG is a known reader of M6A. In future slides, we will talk about how HNR&PG has previously been shown to be gestationally regulated. We will also further discuss HNR&PG's involvement in alternative splicing.
So now let's take a look at HNR&PG expression in pregnant human myometrium. As you can see from the image, HNR&PG is very highly upregulated in pre-non-term laboring uterine sample. However, by term labor and term non-labor, HNR&PG expression is definitely much lower.
Next, let's move on to discussing ER delta 7 and its role in regulating uterine myocyte contractile responses. First of all, ER delta 7 is one of the 3 major ER alpha splice variants that have been identified in the human myometrium. The others being ER alpha 66, which is the full-length isoform, and ER alpha 46. ER delta 7 specifically is a result of skipping exon 7 in the gene sequence. Functionally, ER delta 7 functions as a dominant negative, meaning that it blocks contractile-associated proteins like connexin 43. The image on the slide shows a collagen contraction assay. Looking at these 2 petri dish images, you can see that ER delta 7 expressing cells contract way less than ER delta 7 non-expressing cells. This demonstrates that that overexpression of ER delta 7 inhibits connexin 43 expression and disrupts the uterine myocyte contractile ability. Next, let's take a look at how HNR&PG regulates alternative splicing of ER delta 7. Just as HNR&PG is a reader of M6A, it is also involved in the generation of ER delta 7. From the diagram, we see that if we knock down HNR&PG in human myometrial cells, ER delta 7 is also knocked down. However, ER alpha 66, which is a full-length ER alpha isoform, stays the same. These findings further demonstrate that HNR&PG regulates alternative splicing of ER delta 7. Now let's look at the relationship between MET-L3 and HNR&PG. As I mentioned, HNR&PG is a known reader of M6A. MET-L3, on the other hand, is a writer of M6A, meaning that it places the M6A marks on RNA. After performing the Western blot, we see that MET-L3 expression oscillates during mice gestation. Between days 14 and 17, we see an increase in expression and a substantial decrease by day 18, a considerable increase by day 19, and an absence in expression during labor. On the other hand, with HNR&PG, we see that its expression is more or less the highest between days 14 and 17 of gestation. However, between days 18 and 19, we see a pronounced decrease in its levels. Based on this diagram, we see that there's no direct relationship between MET-L3 and HNR&PG. This is because MET-L3 has other functions besides writing M6A. For instance, it plays a major role in cap-independent translation. So it may not be the best check for M6A expression during gestation. If in the future we find that other writers in the writer complex, including MET-L14 and WTAP, have no other function except writing M6A, it will be better checks to determine M6A expression during gestation. Next, we added tunicamycin to human myometrial HTERT cells. Tunicamycin stresses the cells, so we added it to determine if it had any expression, had any effect on the expression of MET-L3 and HNR&PG. In the diagram, we can see that under stress, so when tunicamycin is added, MET-L3 expression and HNR&PG expression are both upregulated, meaning that they both cause an increase in M6A methylation. Now I'm going to hand it off to Layla, who will talk about the RNA extraction protocol and what we did for a majority of our internship.
So before I go into the protocol, I'm just going to talk about some of the materials that we use. So when I'm speaking about them while I'm presenting on the protocol, you kind of know what it is. So first we have the RNeasy mini kit, which is where we get all of our buffers for the washing and our different tubes. Then we have our centrifuge, which is something we use a lot during this process, which pulls down a lot of our samples that we need. Then we also have the nano drop machine, something that we use to measure the concentrations of our samples. We have our gel, which is kind of like in the middle, and that's just what it looks like when it's loaded. We have the gel documentation machine, which is how we look at our gels once they have ran. And then we also have some examples of a spreadsheet that we developed, so we know how much of the sample and of water that we're putting into the well when we are loading the gel. So what we performed was an RNA extraction protocol using RNeasy mini kit. Approximately 30 milligrams of our animal tissue, which in this case came from the mouse uterus gestational series, was homogenized in RLT buffer with a K-trope. The RNA was bound to a column and then washed multiple times and eluted with RNase-free water. So after this whole process of the RNA extraction, to figure out the concentration of each sample, we use the machine I was just talking about called a nano drop. And this basically tells us the exact concentration for each sample, only using 1 microliter of the sample. So once we figure out the concentration that we have for each sample, we are going to develop a spreadsheet so that we can figure out how much of the RNA sample we are going to be putting into our agar gel, and also figure out how much water we're going to be putting in as well. So for our agar gel, once this is complete, we run the RNA samples on an agar gel to determine the quality of the RNA. And the quality of the RNA depends on the 28S and the 18S bands seen on the agar gel. So now we're going to talk about seeing the bands. So we extracted RNA from 3 different series of mouse uterus gestation tissue. To quantify the RNA, we measured the optical density, or the OD, at the wavelength of 260 nanometers. So we found on some of our extractions that the RNA was degraded, and both the 28S and the 18S bands were not present. As you can see here, the gels are very streaky, and what we want is to kind of see the bands. So we used a E11 as a control because we knew in a previous lab that we did that it was a high quality sample of RNA from a previous RNA extraction that was run. And so we knew that if we loaded, so we allocated the E11 from the previous lab so that we could just use it, so it would be ready to load into the wells, and we wouldn't have to go through the whole process of extracting that RNA over and over again. And so we knew that if the E11 became streaky, that indicated that something was wrong with the loading process, like you see in the first gel. But if we loaded the E11 and it came out fine, like the second gel, you can see there's 2 separate bands, but the rest of the wells are streaky. We know that there's something wrong with the washing, or something probably wrong with just the whole RNA extraction that we did. And so we did have some trials and errors in the beginning, but practice makes perfect. And so we proceeded with the next extraction, and we did so with a lot of caution. We literally banned everybody from our workspace, wouldn't let anybody come in. It was on lockdown. We made sure we sprayed and we RNased literally everything, so nothing would have RNase on it, our workspace, our instruments, or our solutions, and made sure they were all RNase free. And so then the next 3 extractions showed up like this. Wow. Some really beautiful bands. You can clearly see everything is separated, the 28S and the 18S, and this proved that these samples had high quality RNA.
Yeah. To detect changes in epitranscriptomic marking of M6A in mRNA methylation during mice gestation, we use the M6A RNA methylation quantification colorimetric kit to run an ELISA. Before we even ran the ELISA, we needed to have good undegraded RNA samples, so that's why we ran the RNA extraction protocol. So to begin the ELISA procedure, we start with binding the RNA we extracted from the mice to the bottom of these strip wells, as you can see in the second image, using a binding solution. We then use capture and detection antibodies to detect M6A, and then use an enhancer to magnify that detected signal. In order to colorimetrically quantify the absorbance, we add a developer solution to the assay wells so we see a blue color development, and then a stop solution to stop the enzyme reaction. So once the solution turns from a blue color to a yellow color, we can then read the absorbance by using a spectrometer at a wavelength of 450 nanometers. Throughout the procedure, it is important that we wash the assay wells multiple times to wash off any unbound RNA, any remaining capture and detection antibody. The ELISA protocol allowed us to measure the M6A gestational changes quantified to nanogram quantities using a diluted positive control of a known M6A concentration. So these are the results that we got after performing our ELISA. Using the linear regression graph we created from our data, we created a bar graph relating M6A RNA concentration to each gestational day. The optical density from the linear regression graph was changed to nanograms of M6A RNA because the concentration of M6A is proportional to the optical density that was measured. From our data, we can see that M6A concentration is highest on day 15. We also see that the concentration at day 15 is almost double that of the concentration at day 13. However, the M6A concentration then drops at day 17 and stays pretty much that way till term labor. Originally, we hypothesized that we would see the largest increases in M6A concentration during the days where the uterus stretches the fastest. This data supports our original hypothesis because as the uterus quickly grows from days 13 to 15, as we showed in the intro slides, M6A concentration correspondingly increases. In conclusion, our findings implicate that M6A is gestationally regulated. From the ELISA results, that we see that M6A concentration is highest at day 15, which correlates to the point where the mice are growing at their fastest. We also found out that when we added tunicamycin to human uterine cells, both METL3 and HNR&PG expression are upregulated. From this, we propose that ER stress regulates M6A. In the future, just as we did with mouse uterine tissue samples, Dr. Surya is planning on performing RNA extractions and the ELISA procedures with human uterine tissue samples to eventually perform a MiSeq. The MiSeq is an immunoprecipitation using antibodies and RNA fragments that are later cloned to be sequenced. Using the MiSeq instrument, Dr. Surya is planning on sequencing the things that are attached to the M6As. Like I said before, METL3 has other functions besides writing M6A. So it may not be the best check for M6A. So Dr. Surya will also study other writers in the writer complex, including METL14 and WTAP, to see if their only function they have is writing M6A. If it is, they will be better checks than METL3 to determine patterns in M6A expression during gestation. Before we end our presentation, we would like to take a minute and thank some of the people here today. To begin, we would like to thank Dr. Surya, Dr. Condon, and Francis for teaching us so much during our internship these last 8 weeks. Not only did we learn a lot regarding research, but we also learned a great deal about a variety of lab protocols, the importance of having uncontaminated samples, how to use certain instruments, and how to analyze data, and much, much more. Most importantly, however, our mentors showed us that it was okay to make mistakes and have failures. Like Layla mentioned, our experience with the RNA extractions showed us that we shouldn't give up because negative data is still data. It was because of that data that challenged us to think about what went wrong in our procedure and allowed us to make changes before we ran the experiment again. It was after running the experiment multiple times that we eventually got satisfactory results. Next, we'd like to thank all the mentors that spoke to us during our Wednesday lectures. We really appreciate all the advice you gave us regarding our future careers. Next, we'd like to thank Alyssa for organizing all the fun activities during our relationship and internship. Oh,
it's okay, you have a relationship.
During her internship. She made sure to take lots and lots of pictures, so thank you for that, Alyssa. We will never forget the amazing time we had here. Finally, we would like to thank Dr. Moore for starting this program at Wayne and for giving us the opportunity to partake in such an amazing experience. And that's it with our presentation. Thank you for your time, and we hope you enjoyed it.
I'm sure they not have questions for you. They have questions? You're very kind with you. Very good. Let me share a secret with you guys. Don't tell anybody, okay? They are not listening. I go to a lot of, I used to go to a lot of scientific meetings. And 80% of the lectures, I would fall asleep. With you guys, I was awake all the lectures. So I don't know what you did, but it was fantastic. Let me remind you to the parents and the teachers and everybody that the presentations we hear today are coming from high school students. And what you did is just amazing. It's just amazing. Yeah. The clarity of your presentation, the organization of your presentation. And you remember when we met the first time and I asked you, what is your project about? and you couldn't even pronounce uterus and the difficult words, and now you pronounce better than I. So I really want to congratulate you for what you've done. And let's give him an applause because again, I want to reiterate my thanks to all the mentors, the faculty. You did a fantastic job. This is what it keeps us going. Now I can't retire because I know that the future is secure. We have great scientists that will come and we do what we fail. I love your last presentation also. Failure is what it leads to the big discoveries. So you are in the right path, but you're going to have great discoveries. And weekends are open here. You can always come back on the weekends, yes? So, and again, of course, I want to reiterate my thank you to Alyssa for the amazing work. Sharon Pep, I don't know if she was here early in the morning, but Sharon is behind the scenes also helping us to get all the paperwork, permissions. Believe me, to have you here, it requires a lot of permissions from the university, trainings, and so on. And Sharon was instrumental in doing that. Thank you to everybody for coming today. And for those who are in the Zoom. Good. Bye.